Welcome, everyone. Thanks for joining this new session of the ABC seminars. Today we have with us Professor Ole Jensen. He's the chair of Translational Cognitive Neuroscience at the University of Oxford, where he also leads the Neural Oscillations Group. And today he will present some MEG findings on the neural mechanisms of spatial attention and working memory during natural vision and language comprehension. Without further ado, Ole, thanks a lot for being here. The stage is yours.
Yeah, thank you very much. So it's a great pleasure to be here. So I used to work in a low temperature lab back in 1999 to 2002. And back then I was one of you sitting up there listening to talks. So I'm also happy to see there's a lot of old friends still from back then around here. So for me, this was a really great time. It was sort of my formative years for deciding what research I should do in the future. And of course, I learned how to do MEG here and have sort of stuck with that, and being involved now in setting up MEG laboratories, first in Nijmegen in Holland at the Donders, and then at University of Birmingham. And as you just heard, now on Oxford, where there already is an MEG system, but now in the process of establishing an MEG lab. So I'm also going to tell you a bit about that later. But what I'll do now is to tell you about some of the work we have been doing in recent years. And we have been working on how to take some of the basic concepts and mechanisms that we have uncovered in cognitive neuroscience and then apply it to more natural settings, that being reading and visual search, and also working memory in language comprehension. So when starting to think more about how we can apply these notions from cognitive neuroscience to real life settings, there's the issue, and that is, like, often when we do, say, a spatial attention test, we tell people with a cue to attend to the left. A few seconds later, we show a stimuli and so forth, right? So this is all well and good, but of course, in real life, things are going a lot faster, right? So in real life, we are saccading all the time, and for instance, it's clear that we are saccading 3 to 4 times per second, right? So that means that we are doing 150,000 saccades per day, but often saccades are being ignored in the research we are doing on, say, spatial attention and visual search, right? Of course, some people are looking into it, but often it's not really sort of being used as a constraint. And my point is that due to these fast timescales in real test settings, we also need to think about what can sort of be done respecting those timescales in terms of neural mechanisms. So I will move into one of the first studies we have been doing on visual search. And there's somehow a conundrum, right? In a sense that our eyes are moving around from object to object in a visual scene, but typically our eyes land on the important parts of the visual scene. So how can that be, right? How can we move our eyes, for instance, to the man to the right, if we have not really fully processed what's going on there? Of course, that applies to some sort of perifoveal previewing of things out there in the periphery, and that's something we would like to know more about. But there's also a timing issue associated with this, right? So here's an example where you might be scanning the picture in front of you. So first your eyes move to the woman, to the boy, then the woman, and then the dog, right? So now when you fixate on the woman and have to move your eyes to the dog, not only do you have to process the woman, you also have to integrate that image into the scene, but you also have to prepare your next saccade to the dog, for instance. But in fact, there can be several alternatives you can saccade to. And then you also have to plan the saccade, right? All this has to happen within 250 milliseconds, right? So a lot has to be crammed into this time window. So how can the brain do this? So to uncover this, there's a study here by Camille Faxe, where she did MEG, the 306 channel system that was developed here together with eye tracking. And what we did was to present images arranged as you see there on the screen. And these images, I think we had like 4 or 5 categories. and then the participant was moving around to these different images. And what we wanted to ask is that how much can you actually decode of images out there in the peripheria before your eye moves to this, right? It turns out that we don't know too much about this, and we don't know how fast that can be done either. So there was also a memory task, but that we can ignore for now. So what we also were using to investigate the data was to use multivariate pattern analysis, right? You might all be familiar with this now. So it's a technique first developed for functional MRI, and more recently has been used a lot for MEG and EEG data, right? And it works like a charm. So the idea is that we are actually able to decode from the brain activity what kind of category people would be processing, right? So it could be that we train a decoder to tell fruits from animals, and then we show people a lot of fruits and animal pictures, record the brain activity, and then we train a classifier. And then by looking at the distributed activity, we can then decode to what extent people were looking at one or the other category, right? So it's simply the sort of the distributed activity. So a fruit would have a certain brain pattern, and a dog would have another brain pattern, and then we can correlate the activity from the sensors with that template, if you will. And that allows us to tell whether people were looking at one or the other item. So we can do this for categories, but we can also do it for other features. So we can also do it for color versus black and white pictures, right? So that's also possible. So we did this study, and what we had images of 5 categories that we tried to decode, and we also had images that were black and white and in color. And then we simply allowed people to move their eyes around to explore these images. And what we first focused on was the decoding during the foveal processing, as you see here, the foveal processing. And then we were also interested in the decoding of the objects people were about to move their eyes to, right? So first, when we look at the foveal processing, like what people fixate at, as you can see, at 145 milliseconds, we can decode the category of these objects, right? So this is old news. This has been done many times before, and it can be done very reliably. We can also decode the color, meaning black or white or color, and that happened a bit earlier, right? So this is all what you expect. First, we sort of decode the feature of the image, and later on the category. So the big question here is, what can we do in terms of decoding of objects that people are about to move their eyes to, right? So now it turns out that we can do this. Of course, it's not as robust, but at 160 milliseconds, we have a peak in a decoding of category, and slightly earlier, a peak in a decoding of the feature of those upcoming items, right? So there's a debate to what you actually process out there in the paraphobia, right? Some people might doubt that you can decode those at the category or semantic level, if you will. Others say you can, but here now we have evidence for that it's possible to do the category decoding. We can also decode the objects that people were coming from, but I will ignore that for now. So now when looking at the timing, then what you should appreciate here is that in terms of color processing, first, the object you fixate at is decoded at about 70 milliseconds, and at about 90 milliseconds, you decode the parafolia object. And the same when it comes for the category, then it's about 145 milliseconds, and then at 160 milliseconds out there in the paraphobia, right? So there seems to be something going on, which is somehow sequential, but also going on overlapping in time. So then you might ask, is these objects in the fovea and the paraphobia, are they processed in serial or are they processed in parallel, right? So it turns out we cannot get a clean answer to this. In a sense, there's this overlap and still there's some sequential elements. So it's a bit of both, right? So how to think about this? And of course, this is important because there's so much going on within an interscale interval, and multiple objects have to be processed at the same time. So how is that achieved? So what we have been thinking about is that there might be some sort of pipelining mechanism going on, right? So by pipelining, just think about this couple here doing the dishes. So the woman, she's washing the plate, and the guy, he's then drying the plates, right? So of course, these plates are being passed on from one person to the other. So these two people, they are working in parallel, but each of them are doing sort of a serial processing action, right? So this is also what we think might be going on in the visual system. So you can think about before doing the color processing, and then that object is being passed on to for doing object processing, if you will, or category processing. When the object is passed on, that leaves resources for the next object, meaning the peripheral object to be processed, right? So we have tried to put this scheme into a schematic model, as you see here. So there's a lot of details to this, but the key point is that we think that this pipelining process might be going on in a visual system. We still have to find more evidence for it, but we also believe that brain oscillations in the alpha band are involved in clocking this processing. And then you might say, isn't that a bit surprising because alpha oscillation is suppressed during visual processing? But I'm going to get back to that. But so I'm not going to go into too much details here in the interest of time, but as you say, like we have a schematic model out there for what we think is going on in terms of the pipelining process that we are working towards confirming or falsifying. So just to conclude the first part here, so both feature and category of upcoming visual objects can be processed, right? And we can do the category processing before we saccade to these upcoming objects. And we see some element of sequential processing, but there's also overlap. So that means that we are arguing for this pipelining process. Okay, so this was the first study I showed you, and what I'll now show you how we can go in and use some of these ideas and tools to investigate reading. So of course, reading is not too different from visual search in the sense that your eyes are moving from word to word in this case. And there's also a debate to how much is being processed of the words out there in the periphery before your eyes move to them. And of course, reading is a very important task that we all have to learn. So now I'm going to say something that's not terribly surprising, but of course it's the same hardware supporting visual search and reading, right? And also if you look at the brain regions being involved in the ventral stream, is more or less the same brain regions. So now it turns out if you go to Society for Neuroscience, that you'll find that the people studying visual processing and the people studying reading are in very different parts of the poster group, right? So that means that we have two communities investigating reading and visual processing, but they don't really communicate much. And I think that's a bit unfortunate because I think we can actually sort of map on a lot on what we know from cognitive neuroscience to also understanding what's going on in the brain in terms of reading. So in terms of reading, of course, our eye moved from word to word. There's also this notion of doing peripheral processing, that we process a word somehow before our eye moves to it. So what is the evidence for this? Well, the evidence is that sometimes we are doing skipping, right? So if you consider this sentence here, the quick fox jumped over the lazy dog, right? So in this case, the is being ignored, right? So how can you ignore a word when you're moving your eyes? Well, you can only do this if you do some pre-processing of that word, right? So now it turns out there's a big debate in the reading literature on how much is being processed out there in the periphery. So to investigate this, what we have been using is MET again. We also developed a tool that we call rapid invisible frequency tagging. So just to introduce that methodology. So you're probably all familiar with sort of conventional frequency tagging, right? And at least back in the days here in it displays quite some work was also done using frequency tagging. So the idea is that you have different objects in a visual scene, and you can flicker them at different frequencies. And lo and behold, you can measure the brain response from this, right? So now it turns out that you can also do this frequency tagging at higher frequencies, like 60 hertz. So in fact, the brain can sort of keep up pace up to about 80 hertz. And then you don't get a neural response anymore. But it turns out that we can do frequency tagging at 60 hertz, for instance. And this has several advantages. And one being that you don't see the flicker, right? Because if you flicker at 10, 15 hertz, it's pretty annoying. So you get an almost invisible visual image in regard to the flickering. It also gives you a fast time resolution, right? So if processing is turning on and off, and you investigate that turning on and off with a fast sampling signal, meaning the 60 hertz, you can investigate the time course. So and it turns out that we get quite a strong response. I shouldn't say that. We get a response with a high signal-to-noise ratio at 60 hertz. So the reason being that you're probably all familiar with the sort of 1/f of the spectra of the data, right? So up there at 60 hertz, there's not so much noise, but we get a sensible response from the neurons. And we have also been able to demonstrate that if you allocate your spatial attention, you get a stronger frequency tagging signal. So we have been using this technique to investigate reading. And the thing you should imagine here is that we, in this case, are frequency tagging the word fox in this sentence, right? And when doing that, what you should observe is that we get a reasonable frequency tagging response. So this is what you see on the y-axis, right? That is response from the brain in the 60 hertz band. And n refers to fox in a sentence, and n+1 the next word, and so forth, right? So that means that when you fixate on brown, but you flicker fox at 60 hertz, you get the strongest response. However, if you are moved past fox and fixate on jumped, you get a somehow smaller response, right? So what this here demonstrates is that there is this spotlight of attention, if you will, that is moving ahead when you're reading, right? So this is probably not a big surprise, but it shows you that we can measure the attention allocation during reading. So what Yali Pan here has been doing is to design a study where we look into the semantics, if you will, associated with this peripheral word processing, right? So when we talk about semantics, we mean the representations associated with fox, sort of going associated with the meaning of the fox, right? So we all know a fox is a red animal living in a forest, and we also know how to integrate that into word sentences, right? So it's possible to have words that are congruent or incongruent with the sentence context, right? Much like you do in 400 type of paradigms to investigate semantics. So what we have here is two sentences. So the first sentence doesn't really make sense, right? Lily says this blue brother, right? So blue brother, brother in this case here is unexpected with the sentence context. The lower sentence, blue jacket, jacket could very well fit into this sentence context, right? So we have constructed these sentences, and we have some target words that we flicker at 60 hertz. I should also say these words could not be predicted, right? So we made sure that we didn't have predictable words being the target words. And what we then can do is that we can measure the frequency tagging response with respect to the target word. But what is really important to us is to measure the frequency response with respect to the pre-target word. So when people fixate on blue, how strong is the response from the upcoming target word when it's congruent versus incongruent with the sentence context? So when we do this, what we find is that we get a sizable frequency response. So that's what you see here in the time frequency representation. So time 0 is when you fixate on blue, and this red blob here corresponds to the 60 Hertz response from the word brother, right? So when we compare these two representations, we find a difference between the incongruent and congruent response. Right? And in fact, we see that there is a lower frequency tagging signal for the incongruent words, right? So this is a surprise to us because you might have thought that the incongruent word would attract more attention, but it's almost like it's repelling the attention, right? Nevertheless, this is evidence for people doing semantic processing of parafoveal words. For those of you who know the eye tracking literature, it turns out that there is a big debate on this. So I'm not going to get into details, but it has been a decade-long debate to what extent you can extract semantic information from the parafovea. Now we have neuronal evidence for this being the case. You should also appreciate here that this semantic effect occurs very early, namely at 100 milliseconds after 100 milliseconds in regard to when you fixate on blue, the pre-target word, right? So already when you fixate on blue, after 100 milliseconds, there is a neural signature of the congruency of the word in the parafovea. So when you think about the in 400 paradigm, then this is a bit of a surprise, right? So the in 400 response might start to increase after 250 milliseconds, sort of reflecting the congruency or incongruency when you see a word-by-word presentation, right? So what we think is going on here is that you start accumulating information about the semantics of the words in the parafovea, not only one word back, but several word back, right? So that only by that we can explain this very onset of the parafoveal processing. So this is also super important because it also partly explains how we can do so much within one intersaccade interval, namely both process the word we fixate at, but also the upcoming words. Another important factor of this here is that now we have a measure for the parafoveal processing, semantic parafoveal processing, and that's the difference between the blue and the red line, right? So now we can correlate that difference over multiple participants. And this is what you see here. So the y-axis reflects the reading speed, and the x-axis this difference in frequency tagging. So now it turns out that people out to the right in the figure are fast readers, and they are also the ones with a stronger parafoveal effect, and the opposite for people in the left side of the graph, right? So this here actually suggests that fast readers are also the ones who are able to derive the parafoveal information better than those who cannot, right? So it matters. So I'm going to conclude this study that now we have evidence for semantic parafoveal processing, and also this effect is predictive of reading speed. So what I'll do now is to move on to a different study where we are trying to make the same case, right? So when we did the visual search, as I mentioned before, we use the multivariate pattern analysis for uncovering the parafoveal processing. The question is, can we do the same now with MEG doing reading? So this would be a complementary way of looking at parafoveal processing as compared to doing the frequency tagging. So this is a study by Li Jun Wang, where she again presented sentences that people were reading naturally. And what we then did was to do a manipulation where we had pairs of sentences where there was overlap in orthography. So here, if you consider police and policy, they mean very different things, but they have a big overlap in terms of letters, right? So what we now can do is that we can look at the brain activity when people are fixating on clever, and then look at the brain activity and then look at the correlatedness in terms of topography. So we do this simply by doing a correlation over our 306 sensors with respect to word pairs with similar orthography as compared to word pairs with different orthography, right? We can do the same with police and guards. So that reflects more the semantics, the meaning of the word. So police and guard is somehow related concepts in terms of semantics. Again, we can look at the correlatedness of the brain activity. We can ask how similar are the topographies when people fixate at clever, and then do the peripheral processing with respect to these target words. So as I mentioned, we do simply this by doing the correlation over the sensors, right? And this is a technique we developed back in the years with Lin Wang and Gina Kuperberg for looking into prediction during language comprehension. Now it turns out that this pairing of the words also works very well when doing reading. And the first result is here, where we consider police and policy, meaning orthographic overlap, right? So it turns out that when people fixate on clever, and when we look at the correlatedness in the brain activity, then about 80 to 100 milliseconds in, we see an increase in the correlation for the orthographic pairs as compared to pairs that do not look similar to each other. So that's the between pairs, right? And also for this effect, there was a correlation between the orthographic effect and reading speed. So this is probably expected, or I should say we could also localize this effect. So we did a searchlight approach, and then we found that this correlatedness was strongest in the visual word form area. Now, when we do the same with respect to semantics, like police and guards, and consider when people fixate on clever, then a bit later we find a difference in the correlatedness, right? And this difference in correlatedness also correlate with reading speed. So this here is complementary evidence for people processing the meaning or semantics of words already before their eyes move to those words in the paraphobia. And we can localize the effects with our searchlight approach. So for semantics, there is a Broca's area coming up in the left prefrontal cortex. And then there's also a right sort of parietal occipital source. So this right source, we don't really know what to think about, but this left inferior frontal gyrus source is probably somewhat expected. One might also have expected something going on in the anterior temporal lobe, but we didn't see this. But of course, the left inferior frontal gyrus is sort of a classic language area. So to conclude this part, we now have evidence for both orthographic and semantic peripheral processing as uncovered by this multi-pattern analysis, and both effects for orthography and semantics are predictive of reading speed. And again, we find the semantic effect sort of 120 milliseconds in after fixating on the pre-target word. Good. Now I mentioned this pipelining mechanism before for visual search, and we think a similar mechanism might be going on during reading. So in this case, you can think about one region doing the processing for the visual word form area, and then there's a downstream region doing the semantic processing. And again, the words are sort of being shifted between these regions in a pipelining fashion. And we have also made that explicit in a model. So this is what you see here. First, the word is being processed for orthography, then you move on for lexical and semantic processing. After that happens, the word in the paraphobia over can then be processed in the visual word form area and so forth. So everything is sort of being shuttled down through the ventral stream in this manner. And again, we have a schematic model for how this is going on, right? So as I mentioned before, we also think that brain oscillations are involved for doing this clocking. So it's part of a bigger scheme in which we have found that alpha oscillations are pulse inhibition. They reflect pulse inhibition, right? So these alpha oscillations are about 10 to 12 hertz. What we've been able to demonstrate on others is that they indeed are inhibitory, right? But of course, they're rhythmic too. So you can talk about rhythmic pulsing. So the core function of these oscillations, we think, is that they, at the peak of inhibition, suppress everything. But as the inhibition ramps down, the most excitable item is then activating, and then the second most excitable item. And that actually means that if you have competing words like jumped and over, both being within your visual field, you first process jumped as it overrides inhibition and then over, right? So you can get this conversion of parallel input to a sequential processing stream by these releases of inhibition by the oscillations. And this is actually an idea we have stolen from the rat hippocampus, where you see something very similar in terms of processing of hippocampal place cells. Nevertheless, of course, this here also presupposes that alpha oscillations are there during visual processing, right? But this process here also then predicts that there should be a link between saccades and the alpha oscillations. So the idea is that when you move your eye, if you can do this locked to the phase of the alpha oscillations, it would be such that you move your eye so the information comes in at the, say, excitatory phase of the alpha oscillations. Of course, this is a very strong prediction that we can test, right? It also solves another conundrum, and that is if you think about the alpha oscillations being 10 hertz, right? And this is sort of a clocking process for the visual system, then it is a bit odd that you move your eyes 3 to 4 times per second, right? So from a design perspective, it would be weird to design a brain with a clocking frequency at 10 hertz if you're also doing a sampling of the environment at 3 to 4 hertz, right? So the only way to solve that conundrum would be if there's a link between the saccades and the alpha oscillations. And note here, I'm not going to suggest that the saccades are occurring at every alpha cycle, but what I'm saying is that at each alpha cycle, there's a window of opportunity for making a saccade, but it doesn't have to be in every alpha cycle. And just to show that this is not completely crazy, here's a study by a Japanese group in the marmoset monkey where they do ECOG recordings. And what they find is that each time the monkey is doing a saccade, you get a propagating wave in the 8 hertz band, sort of the lower end of alpha that goes from sort of frontal to posterior in the dorsal stream, and then it goes frontally in the ventral stream. So it's the same kind of alpha wave we think might be carrying and organizing the processing during reading. Anyhow, to investigate this, what we did was to take our previous reading data. We have the eye tracker, we have the ongoing MEG, and we can now look at these oscillations during reading, right? And some of you might be puzzled about this alpha talk, because of course for a good 100 years there's been the notion that alpha is being blocked during visual processing, right? So lo and behold, if you flash a big image in front of a person, you do see a suppression of the alpha oscillations. However, it turns out that if you allow people to do visual search during reading, these alpha oscillations actually remain relatively strong. Not as strong as during rest, but they are still often the strongest rhythm that you see during that you see in the power spectrum. So what you have here is a time frequency representation of power And it's this time frequency representation are aligned to saccade onset, right? And what you should appreciate here, that except for the 1RF, the alpha oscillations are still the strongest. What we also see that the topography here is that there's a clear posterior topography. And then we also see a left-right source, right? So in different kind of motor tasks, there's been the discussion. So Rita has been working on this, or has worked on this decades ago, right? Where are the sources of this more frontal alpha oscillation or mu rhythm, right? And there's been some discussion to whether it is in motor cortex, in premotor cortex, in motor cortex, primary motor cortex, or somatosensory, primary somatosensory cortex. But we actually start to see now is that during reading and visual search, these sources start to overlap with the frontal eye field. I think I'll show you that in a minute. But nevertheless, the key point is that we have strong alpha oscillations during reading and visual search. And what we now can do is to look into the phase of these oscillations. And the way we do that is that for each saccade, we go in and cut out a snippet of data, right? So the upper trace here is an example of the eye tracking. Then we cut out snippets of data, and then we do a phase logging analysis with respect to the saccade onset, right? So this is done by using the phase logging factor or intertrial coherence. And when we do this, what we see is phase logging in the alpha band prior to the saccade, right? I should also mention that we see this being stronger when saccading to low versus high frequency words, right? But that's a bit of a side story. The key point being that often we see that the saccades are locked to the phase of the ongoing alpha oscillations, right? And since we see this phase locking prior to the saccades, then this also suggests that the alpha oscillations are the boss, right? They are in control. So it's the phase of the oscillations determining when you can saccade or not the other way around. So then you might ask, was this also the same in our visual search study that I showed you first, right? And we can do exactly the same analysis. Again, we see strong alpha oscillations during visual search. And in this case, we have been able to localize the frontal sources. And indeed, we do see sort of quite widespread sources, but they do overlap with the frontal eye field, right? So what I'm getting at is that I also want to involve the frontal eye field in this process of controlling the alpha oscillations. And again, during the visual search study, we see this intertrial coherence or intertrial phase coherence prior to the saccade onset. And we also have a third study done by Tobias Staudigl back in the days where we see this locking of saccades to alpha oscillations, right? So it's just to say, like, this is not sort of a small random finding that's hard to replicate. We now have 3 studies, both during visual search of people searching to different objects or natural scenes, or during reading, where we see this locking of the ongoing oscillations of the saccades to the ongoing oscillations. So to me, this also sort of forces a rethink of what alpha oscillations are doing, right? So they have been thought to be sort of a resting rhythm or inhibition rhythm of the visual cortex. But what we would like to think now is that these oscillations are actually involved in doing the visual motor coordination. And also the frontal eye field is supposedly very strongly involved in this. So yeah, so to make a long story short, we now think that these alpha oscillations they are controlling when saccades can happen, but we would also like to think that they are involved in organizing this pipelining mechanism. For the latter, we still have a lot more of work to try to substantiate this. Okay, so I'm going to use the last part of the talk to talk a bit about what we want to do in the future, right? So What we would like to do is to take what we have learned about reading and then apply that to children learning to read. What we want to ask is, at what stage do children acquire this fluency as indicated by not taking in one word at a time, but to take in multiple words, right? Of course, when we all started to learn to read, we learned to sort of map the text onto the sound and reading out aloud, and we did this word by word. However, as we became more fluent in reading, we also started to take in multiple words at once, right? And that, of course, required doing the perifocal processing. Now, it turns out not so much has been done in terms of acquiring reading skills and investigating perifocal processing, possibly because there hasn't been the tools to do this. But now we have the tools for doing this, mainly using the frequency tagging and the multivariate pattern analysis. So this is what we want to investigate. Furthermore, there's also quite an interesting point about the alpha oscillations. So it turns out then when children are about 3-4 years old, these alpha oscillations are actually in the theta band, maybe 5-6 hertz. And then as children are acquiring reading skills, you also see an acceleration of these alpha oscillations. So if you now think the alpha oscillations are important for coordinating the saccades, then there should also be a link between the frequency of these oscillations and your ability to acquire fluent reading skills. And what we want to do eventually is also try to take that insight you will gain through this to investigate reading disorders. So, of course, one problem of doing reading in children is that the MEG system is too big, right? And the kids are simply bobbing around in the helmet. So what comes to the rescue is, of course, these new sensors, the OPM sensors. Of course, there's a lot of excitement in the field, and many sites have now gotten OPM systems up running. So we have one system in Birmingham. So there we are also lucky to have Anya Kowalczyk. She can develop her own sensors, but we have also a commercial system from Feedline up running. In Oxford, we just got a system installed from Circa with the Q-spin sensors, and that's due to the efforts of Mark Bullrich and Rob Seymour. These OPA systems are now very suitable for doing recordings in children because we can adapt the sensor array to the individual head sizes. And this is exactly what we want to do in the future. So we are in the process of developing the paradigms of doing this. But I should say that these OPM systems, they do work surprisingly well. We get good data, and now we can do our sort of bread and butter cognitive neuroscience studies, at least in adults. So also a shameless plug, we are doing a course on how to analyze OPM data. It turns out it's of course not very different than doing MEG data, but the devil's is in the detail. So we have a course in June where we have developed a pipeline based on M&E Python, where we sort of provide step-by-step guidance for how to analyze these data in the context of a cognitive neuroscience paradigm. So I'm going to summarize here, and what I've been providing you is with data, providing evidence for that it is possible to do semantic processing or processing the meaning of both objects and words in the periphobia. This processing occurs quite fast already at around 100 milliseconds. There is information in brain signals about what's out there in the periphobia. Furthermore, when doing this reading and visual search, it turns out that the saccades in the visual system are phase locked to alpha oscillations. We think that's very important because it forces a rethink about what these oscillations are doing. We have a theoretical work, sort of a pipelining mechanism for how this processing is being organized. So this is still tentative and is something, a framework we are working towards. So in the future, of course, we want to find further support for the pipelining, but we also want to investigate how these alpha oscillations are involved more specifically in doing the circadian control and organizing the visual processing. One way of doing this is also to try to get causal evidence in a sense that it's possible to sort of knock out the frontal eye field with RTMS, so we can reduce people's ability to modulate the alpha oscillations, and it should also impact on how they do circadian and peripheral processing during reading. And finally, we now have the funding also to do these investigations on reading to children. And this is something we are very excited about because we now believe we can take findings from cognitive neuroscience and then apply them to understand how reading skills are developing in children learning to read. So I'll end here and acknowledge the group that has been involved in this work, and thank you very much for your attention.
Thank you very much.